In autism spectrum disorders, we observe a range of physiological and neurological challenges, including motor neuron dysfunction, which often manifests as fine motor impairments, such as difficulties or complete absence of speech. While these issues can stem from various inflammatory processes, one area of particular interest is cyanide toxicity. Anecdotal evidence suggests that cyanide levels may be elevated in autism, with certain gut bacteria such as Pseudomonas aeruginosa and Prevotella acting as potential sources. On the behavioral side, we frequently see self-injurious behaviors, stimming, hyperactivity, and heightened emotional dysregulation, which can often be tracked back to gut-derived neurotoxins, including bacterial endotoxins, ammonia, histamine, and oxalates. These metabolites can profoundly influence neurotransmission, immune activation, and metabolic function, contributing to the behavioral phenotypes observed in autism. Each of these topics we have discussed extensively and can be accessed for free on our Rumble channel, as well as on our website at autismisbiomedical.com. What I wanted to talk about today, however, is something different, particularly finding a neurobiological basis that could explain the extraordinary cognitive abilities despite these dysregulations as mentioned above. It is hard to imagine, but one thing I have learned after 17 years of studying autism is that we cannot judge intelligence by looking at behavior. One particularly intriguing area of discussion is the extraordinary abilities exhibited by some, possibly all, individuals on the spectrum, including exceptional abilities in memory, complex information data, and abstract imagination. As highlighted in the #1 podcast, The Telepathy Tapes, there are even reports of heightened telepathic abilities and an enhanced sense of connection with consciousness. We know this because many severely autistic children who are nonverbal can bypass this blocked speech pathway and are able to communicate their thoughts with typing or spelling. If we consider that neurochemical imbalances in autism may amplify perception, sensory processing, and nonlinear thinking, it could offer a plausible neurobiological basis for these phenomena. Conversely, the same heightened sensitivities could also explain extreme anxiety, panic attacks, and other severe behavioral manifestations in autism, which unfortunately we see a lot of and is the primary reason parents really reach out for consultation. By exploring both the disruptive and extraordinary neurological outcomes in autism, we have uncovered new mechanisms that bridge microbiome influences, neurotransmitter metabolism, and higher cognitive function, shedding light on both the challenges and unique abilities observed in these individuals. A 2010 study out of Italy found significantly elevated levels of bufotenin in the urine of individuals with autism and schizophrenia compared to controls. Bufotenin is a naturally occurring compound that acts on serotonin receptors and has hallucinogenic properties. It is an oxidized form of DMT, which is dimethyltryptamine. The study also found a strong correlation between bufotenin and hyperactivity in autism, and they suggested that altered serotonin metabolism may be driving behavioral symptoms. In my years of testing urinary neurotransmitters, I've consistently observed high levels of tryptamine and serotonin in children with autism, strongly suggesting a dysregulated pathway. This aligns with what we now suspect: a microbiome-driven pathway leading to bufotenin and DMT overproduction. Certain bacteria, particularly Prevotella, Clostridia, and Disulfibreo, are often overgrown in autism and significantly alter tryptophan metabolism. Prevotella in particular has tryptophan decarboxylate activity, meaning it can convert tryptophan into serotonin directly in the gut, bypassing the body's normal regulatory mechanisms. My colleague Alex Zaharikis has analyzed over 2,000 stool samples in autism and has repeatedly seen these bacterial overgrowths in these children. Since 90% of the body's serotonin is produced in the gut, this microbial overactivity can upregulate serotonin synthesis, potentially overwhelming normal pathways. Once serotonin levels rise, the body may compensate by methylating excess serotonin via an enzyme called INMT, producing DMT, an even stronger psychoactive compound than bufotenin. Certain gut bacteria could further enhance this process by increasing methyl donor availability through pathways involving SAMe, folate, and methionine. DMT itself is a hallucinogenic compound, but it can be further hydroxylated by monoamine oxidase, the MAO gene, into bufotenin, which is 5-hydroxy-DMT, which also has psychoactive properties. In autism, where liver detox pathways such as phase 2 methylation and glucuronidation are often impaired, bufotenin may accumulate rather than being effectively broken down and excreted. This could explain why elevated bufotenin levels have been directly correlated with hyperactivity scores in autism. Bufotenin acts on 5-HT2A and 5-HT2C serotonin receptors, the same targets as LSD and psilocybin, which could explain sensory processing disturbances, aggression, and hyperactivity frequently reported in autism. Clinically, urine neurotransmitter testing confirms this pattern: high serotonin, high tryptamine, and likely bufotenin overproduction. Microbiome testing via 16S sequencing requires proper interpretation, and my colleague Alex Zaharakis has done this for the last 5 years. Particularly looking at these bacterial overgrowths, it could provide deeper insights into this metabolic imbalance. This isn't just theory, it's supported by biomedical data. We now have strong evidence that in autism, gut bacteria may hijack neurotransmitter pathways, leading to the overproduction of bufotenin and other psychoactive compounds like DMT, cyanide, and other toxic substances, potentially contributing to the particular phenotypes we observe on this wide spectrum. Understanding this opens the doors for new diagnostic and therapeutic interventions targeting microbiome balance and neurotransmitter metabolism to improve outcomes in children with autism. For more information on what we do, please visit autismisbiomedical.com. We're a nonprofit organization helping families with counseling as well as sponsoring biomedical testing and intervention with our gut balancing program. Please visit our site and read about the amazing testimonials we receive from parents and to learn more on how you can get started and how you can contribute. Thank you.